Good morning, students. Last time we have started topic number 14, biomolecules, in which we have completed carbohydrates. Now we are going to start new point or new biomolecule is protein. Now first we are going to see introduction. Proteins are the fundamental structural material of animal bodies. Proteins in the form of enzymes play prime role in all the physiological reactions. The name protein is derived from the Greek word proteus, which means primary or of prime importance. Nutritional sources of proteins are milk, pulses, nuts, fish, meat, etc. Chemically, proteins are polyamides, which are high molecular weight polymers of the monomer unit called alpha amino acids. That is, the monomer of this protein or polyamide is alpha amino acid. Now first we are going to see alpha amino acids. Proteins on complete hydrolysis give rise to mixture of alpha amino acids. That is, proteins on hydrolysis gives alpha amino acids. Alpha amino acids are carboxylic acids having an amino, that is NH2 group, bonded to the alpha carbon, that is the carbon next to the carboxylic group. Alpha carbon in all the alpha amino acids except glycine is chiral. It is found that the alpha carbon in alpha amino acids obtained by hydrolysis of proteins has L configuration. The L alpha amino acids are represented by the Fischer projection formula as shown in the figure. This is the structure of alpha amino acid RCHNH2COH. Now this is COH group, that is carboxylic acid group, which is bonded to this carbon atom. It is called as alpha carbon atom. This alpha carbon atom is bonded to alkyl group and NH2 group, that is amine group. Now here this amine group is present on alpha carbon atom, therefore it is called as alpha amino acids. This is the example of alpha amino acid. And this carbon atom, that is alpha carbon atom, which is bonded to alkyl group, amine group, hydrogen atom and carboxylic acid group, is asymmetric carbon atom, therefore it is called as chiral carbon atom. And therefore these alpha amino acids are optically active compound. Now here we know that what is meant by L configuration and what is meant by D configuration. We have studied this in carbohydrates. Now here the chiral carbon atom, which is bonded to COH group, NH2 group and R, is alpha amino acid. Now here this alpha amino acid are L alpha amino acid, that is their configuration is L configuration. Now here this L alpha amino acid, this is the structure of L alpha amino acid according to Fischer projection formula. Now here this lowest chiral carbon atom is bonded to NH2 group and hydrogen atom. Now this NH2 group, like OH group in carbohydrates, this NH2 group is present on the left side, therefore it is L alpha amino acid. Now this is the example of alpha amino acid, glycine. In glycine, this alpha carbon is not asymmetrical. That is, it is bonded to two hydrogen atom, one amino group and one carboxylic group. Therefore, it is achiral. That is, it is not a symmetrical carbon atom. The symbol R in the structure of alpha amino acids represents side chain and may contain additional functional group. That is, this alkyl group, which is bonded to alpha carbon atom, is a hydrocarbon radical which contain any additional functional group. If R contains carboxylic, that is COH group, the amino acid is acidic in nature. That is, this amino acid is acidic amino acid. If R contain amino group, either it is primary, secondary or tertiary amino group, then it is called basic amino group. That is, if this alpha amino acid which contain alkyl group attached to alpha carbon atom, this R contains carboxylic group, then it is acidic amino acid. If this R contain NH2 group, then it is basic amino acid. The other amino acids having neutral or no functional group in R are called as neutral amino acids. That is, if R, which is attached to alpha carbon atom, does not contain any functional group, then it is called as neutral amino acid. Alpha amino acids have trivial names and are generally represented by 3-letter symbol or sometimes by 1-letter symbol. Now we are going to study this in the next table. This table lists the 20 alpha amino acids, often referred to as simply amino acids, commonly found in proteins, with their symbols and also their types as neutral, acidic or basic. In that table, 10 amino acids from this list cannot be synthesized in human body and have to be obtained through the diet. These are called as essential amino acids and are marked with asterisk, that is star, in the table. That is, some alpha amino acids can be synthesized in the body and therefore they are called as non-essential amino acids, and some amino acids cannot be synthesized in the human body and therefore we have to obtain these alpha amino acids through diet. They are called as essential amino acids. Now this is the table listed of alpha amino acid. Now there are natural alpha amino acids. Natural alpha amino acid, that is LRCH-NH2COH. Star indicates essential alpha amino acid. Now here first we are going to see neutral alpha amino acids. First is glycine. Now here R is H. This symbol of this glycine is represented by GLY, that is 3-letter symbol. 1-letter symbol is G. Then second is alanine, in which R is CH3. This alanine is represented as ALA, that is 3-letter symbol is ALA, and 1-letter symbol is A. Now valine. Now here this valine has star, therefore it is essential alpha amino acid, in which R is Me2CH, that is 2-methyl group attached to CH. It is valine and it is represented by the symbol VAL or V. Then 4 number, leucine. It is also essential alpha amino acid. R in leucine is Me2CH CH2. 3-letter symbol is LEU and 1-letter symbol is L. Then 5 number, isoleucine. This is also marked as a star, therefore it is a essential alpha amino acid. R in this isoleucine is CH3 CH2 CHMe. It is represented by 3-letter symbol like this, ILE, and 1-letter symbol is I. Then next, asparagine, NH2CO CH2. It is represented as ASM or N. Then 7 number, glutamine. In glutamine, R is NH2CO and CH2 CH2. It is represented as GIN or Q. Then next 8 number is serine, OHCH2. It is SER. It is represented as SER and 1-letter symbol is S. Then next is threonine. Now it is essential alpha amino acid, CH3 CHOH. It is represented as CHR and T. Then 10 number, cysteine, SHCH2. It is represented as CYS or capital C. Then methionine, MESCH2CH2. MET. It is represented as MET and one letter symbol is M. Then 12 number, phenylalanine, pH CH2. Now here the 3 letter symbol is PHE and 1 letter symbol is F. Then 13 number, tyrosine. Now the alkyl group in this tyrosine is para OH C6H4CH2. 3 letter symbol is TYR and 1 letter symbol is Y. Then 14 number, tryptophan. Now it is essential alpha amino acid. This is the structure of alkyl group present in tryptophan and 3 letter symbol is TRP and 1 letter symbol is W. Then 15 number, proline. This is the alkyl group present in proline. This is the entire structure of this proline, which possesses COH group and NH group. That is, it is entire structure of alpha amino acid, and 3 letter symbol to represent proline is PRO and 1 letter symbol is P. Now all these 1 to 15 alpha amino acid are neutral alpha amino acid. Now we are going to see acidic alpha amino acid. Here aspartic acid. Now in aspartic acid, the alkyl group attached to alpha carbon atom is CH2COH. That is R is CH2COH. Now this R possesses carboxylic group, therefore it is acidic alpha amino acid, and it is represented by the symbol ASP or D. Then next one is glutamic acid, COH CH2 CH2. This is the alkyl group attached to the alpha carbon atom of alpha amino acid, and it is represented as GLU or G. Now we are going to see basic alpha amino acid, in which first is lysine, LYSINE. Now it is also essential alpha amino acid. Now here the alkyl group attached to alpha amino acid, alpha carbon atom of alpha amino acid is NH2CH2 4 times. It is represented by LYS or K. Then next, arginine. Now the alkyl group attached to the alpha carbon atom of alpha amino acid is CH2NH, CNH2 double bond NH. Now it is represented as ARG or R. And last one is histidine. It is also essential alpha amino acid. Structure of this alkyl group, which is attached to the alpha carbon atom of alpha amino acid, is this, and it is represented by HIS or H. These are the examples of alpha amino acid. Now we are going to see physical properties of alpha amino acids. Alpha amino acids are high melting water soluble crystalline solids, unlike simple amines or carboxylic acid. These properties are due to the peculiar structure called zwitterion structure of alpha amino acid. Now we know that what is meant by zwitterion. In amines or in the diazonium salt, we have seen what is meant by zwitterion. An alpha amino acid molecule contains both acidic, that is carboxylic group, as well as basic, that is amino group, that is NH2 group. Proton transfer from acidic group to basic group of alpha amino acid form a salt, which is a dipolar ion called zwitterion. Amino acid can exist in different forms depending upon the pH of aqueous solution in which it is dissolved. Consider, for example, zwitterion and the other form of alanine. Now this is the structure of alanine. That is, here this NH2 possesses lone pair of electron and this carboxylic acid group. Now this carboxylic group can donate a proton. Now what happens? The reaction between this basic group and this acidic group is carried out. Now we know that NH2 group possesses a lone pair of electron and therefore it can easily accept a proton, and therefore this carboxylic acid donate this H plus ion to this NH2 group to form NH3 plus and here COO minus. This ion is called as Zwitter ion or dipolar ion. Now we are going to see peptide bond and proteins. Peptide bond and proteins. Proteins are known to break down into peptides. That is, when breaking of protein takes place, formation of peptide takes place in stomach, duodenum, under the influence of enzyme. Pepsin being one of them, which is secreted by stomach. Polypeptides are further broken down to alpha amino acids. This implies that proteins are formed by connecting number of alpha amino acids to each other. The bond that connects alpha amino acids to each other is called peptide bond. Consider, for example, linking of a molecule glycine with that of alanine. One way of doing this is to combine carboxylic acid of glycine with the alpha amino group of alanine. This results in the elimination of water molecule and formation of dipeptide called glycylalanine, in which the two amino acid units are linked by a peptide bond. Now this is the reaction. NH2CH2COH, this is glycine, and NH2CH3COH is alanine. Now here reaction between the COH group of glycine and NH2 group of alanine takes place. Here removal of water molecule is carried out to form CONH linkage. This CONH linkage is called as peptide linkage. This is peptide linkage or peptide bond, and this compound formed is glycylalanine. Now here only removal of H2 from this COH group and H2 group takes place, and this CO group get attached to this NH group to form peptide bond or peptide linkage, and here formation of dipeptide takes place. Name of this dipeptide is glycylalanine, that is NH2CH2 CONH CH3 COOH. If combination of third molecule of alpha amino acid with dipeptide would result in formation of tripeptide. Similarly, linking of 4, 5 or 6 alpha amino acid results in the formation of tetrapeptide, pentapeptide or hexapeptide respectively. When the number of alpha amino acids linked by peptide bond is more than 10, then products are called polypeptides. In this way, formation of polypeptides takes place. The CHR units linked by peptide bond are referred as amino acid residues. Proteins are polypeptides having more than 100 amino acid residues linked by peptide bonds. It may be, however, noted that distinction between proteins and polypeptides is not sharp. That is, proteins and polypeptides are nearly similar. The two ends of polypeptide chain of proteins are not identical. The end having free carboxylic group is called carbon terminal, while the other end having free amino group is called N terminal. In the dipeptides, glycylalanine glycine residue is N terminal and alanine residue is a C terminal. That is in case of this glycylalanine. Now it is N terminal and this is C terminal. That is, glycine possesses N terminal and alanine possesses C terminal.